Hey guys, I am going to give you a little bit of help on the carbon transfer through snails and Elodea. You've been doing these virtual labs, but they've been pretty straightforward. In this experiment, you're actually going to create your own setup, and then you're going to run the experiment. So when you first open the web page or the lab, the problem's going to come up. So what you are trying to solve is you're setting up a snail aquarium at home. So your aquarium kit contains a bag of snail eggs and an Elodea plant. So the instructions say to pour the contents, including the Elodea, into the aquarium, but you're not sure why you need the Elodea, why you need the plant. So what is the problem that you're trying to solve? Well, you're trying to solve why might Elodea plants be important in maintaining a healthy ecosystem. At the end of this unit, hopefully you have a good understanding of cellular respiration and photosynthesis and how those two are related. And that information will help you in this experiment. So what you're going to be doing is you're going to be finding the relationship between plants and animals, or Elodea and snails, in setting up a healthy and balanced aquarium. So that's what you're going to be doing. As you click through each type of, or what you're going to do is you're going to explore the checklist. So these are all of the materials that you're given. You have to click through every single one before it will allow you to move on. I'm going through these pretty quickly, but make sure that you are paying attention to what they are, because they're going to be important as you go through each part of the experiment. So it looks like we have it all, and it's not going to allow you to move on until you've clicked through all of this. So now I'm going to go through procedure, and it's telling me to open the lab notebook. So this is a little bit different. So you guys are going to be creating a hypothesis. So here's your problem up here, and then your hypothesis should explain or should answer a problem. So what you're trying to explain is how carbon dioxide cycles in aquariums, in aquarium water through snails and Elodea. So think back to the equation for cellular respiration, think back to the equation for photosynthesis, and hopefully this information will help you come up with a concise and reliable hypothesis. So remember, you guys, this doesn't have to be correct. This is just what you are going to test. Hypotheses can be reevaluated, restructured, and redone as you go through the experiment. So remember, your hypothesis is just your prediction or your probable solution to the problem. So once you've created a hypothesis, I'm just going to put something else, then you're going to be allowed to move on. Now, in the past, we have been filling out lab papers that look like this, and you're more than welcome to do that. But in this particular lab, it's not going to allow you to move on until you fill in these spaces. So what I recommend is that you actually do the lab within here, and then at the end, you're going to print. And I'll show you how to actually save it as a file rather than print out paper. So as I go through now, all right, so now we're actually going to create an experiment. So here's my hypothesis. Hopefully yours looks a little bit better. It's going to go through your dependent variables, your operational definition. You need to make sure that you're filling in each blank. It's going to go through your independent variable. It's going to ask you what your independent variable will be. So there's a few different things that you can go through based on our lab supplies. So the number of snails, the number of Elodea plants, or the amount of light. So remember, we have a little light, growth light over here. Then we also have this box that can create darkness. So these are all things that you can test, all these variables. Now remember, we don't want to try to do a bunch of different variables because then we don't know which one is going to be changing the outcome of our experiment. So you can create your independent variable. You can also go back and change it. So if you run the experiment and you don't like it, just restart the experiment. There's actually a button where you can do that. Make sure that you have a control, and then it's going to ask you to create a design, okay, the setup for each tube, and it asks you how many test tubes you're going to be using, and just make sure that one of them is your control. And then the other test tubes can be used for your experimental conditions. So whatever you're changing, whatever your independent variable is going to be. Now it's not going to let me move on until I've filled this out. And this is actually where I'm going to leave you guys. I will show you how to print this. So what you're going to do now is it's going to show the work that you've done in the actual lab. So when you're filling this out, it's going to fill it in for you. Okay, so you're going to have all of this data. You're going to answer all of the conclusion questions within the lab. Now, instead of printing to, so normally I would print to my printer and then it would print out and I would have some pieces of paper, but you actually can print it or save it as a PDF, and that's what I want you guys to do. So you're going to save as a PDF. And then you're going to submit that PDF into Canvas. And so rather than go back and forth between here and do the process twice, I just want you to fill it in the lab, save it as a PDF, and submit that. If you guys have any questions or if you need any help deciding on what you want your independent variables to be for this lab, go ahead and email me or call me, and you're also you can also Skype me too. So not a lot of you have done that. So make sure that you're Skyping me if you need some help with questions. It doesn't even have to be on this lab. I hope this helps, and let me know if you guys need anything else. Thanks.